Hello! In this video, we're going to talk about, we're going to have an overview talk about SARS CoV-2 testing. If you're wondering what is SARS CoV-2, it is Severe Acute Respiratory Syndrome Coronavirus 2. Okay, yung COVID, yung nagkakos ng COVID-19, yung dating tinatawag na novel coronavirus. Okay, so the first step in this process is sample collection. Kine-collect ang sample from various parts of the body kung saan nag-gather ang SARS-CoV-2, such as dun sa ilong ng tao o kaya sa may throat niya. Nose, nasal, and then throat, which can actually be the pharynx, so nasopharyngeal. So, yung sample niyan, matitreat siya with several chemical solutions para ma-purify siya, matanggal yung proteins, yung fats. Matira lang is yung RNA na present sa sample. Yung RNA na yun, it can actually be an RNA na mismong genetic material na. It can lead to the genetic material mismo ng tao on kung saan siya nakuha, or pwedeng yung RNA din mismo nung virus. Okay, so importante na ma-maintain yung integrity ng RNA kasi kung syempre hindi siya pure, alam niyo yung mangyayari, self-explanatory. Okay, so it's the main goal of isolating it is to stabilize the RNA. Para rin actually one of the other goals too is dapat kapag ini-stabilize mo yung RNA, ginagawa mo rin is ini-inhibit mo rin yung mga ribonucleases. Kasi kapag hindi mo in-inhibit yung activities ng mga yan, ng ribonucleases, masisisirain. Okay, madadigest yung RNA. So, ayun. We are maximizing as well, as much as possible, yung yield by making proper adjustments sa storage at saka sa extraction methods. Kasi ano lang yan isang maliit na swab lang yan. So, we need to do everything in our power din na ma-preserve kung anumang konting sample na meron tayo. Okay, I hope that is clear. Purification methods, usually ginagawa dyan, tinatanggal natin yung mga compounds tulad ng mga polysaccharides, mga acids na galing sa plant tissues na pwedeng mag-interfere sa activity ng enzyme. And then yung mga covalent inhibitors nga, tulad ng RNases. Ayan. So, even yung mga reverse transcriptase, yung mga salts, yung mga metal ions, ethanol, at saka yung phenol, kailangan din sila matanggal. Kapag pure na talagang RNA yan, i-store siya at 80 degrees Celsius with the minimal freeze-thaw cycles. Ayan. Now, ang objective ng SARS-CoV-2 testing is ma-identify yung parts ng viral genome, yung DNA, dun sa patient sample. Kailangan makikita yon. Okay? Kasi pag di nakita, ibig sabihin nun, negative yung result. Okay, yun yung objective ng testing. Kailangan makita. Kapag nakita yung part ng viral genome sa pasyente, dun sa sample sa pasyente, ibig sabihin nun, infected siya. Kapag hindi nakita, ibig sabihin nun, hindi siya infected. Ngayon, anong bang part nung viral genome pinag-uusapan natin? It can actually be different sources, of course, are going to tell us na ito yan. Pero usually sinasabi nila is yun yung N gene. Okay, so yung viral genome, kapag meron siyang N gene, ibig sabihin nun, infected yung tao with coronavirus. Ayun, so I hope you get that. Now, meron ka na RNA. What do you do next is you do reverse transcriptase polymerase chain reaction. Okay, so let me tell you again na konti nga lang yung sample na meron tayo. Okay, so there is a need para there's a need for more samples, for more copies of the RNA. So, anong gagawin natin yan? Yung RNA na na-detect from the sample, patient sample, ang gagawin sa kanya is lalatagan siya ng mga primers. Yung primer niyan is mare-recognize niya yung mga specific RNA sequence within the viral genome that bracket the region of the N gene. Okay, so yung mga primers na ilalagay mo dyan, nare-recognize nila yung N gene. Remember, ano kasi yung N gene? Ito yung part ng genome na kapag present siya, infected yung tao. Correct. Eh di ba yung RNA na yan, di ka pa naman sure kasi mixture yan ng RNA ng tao at saka yung RNA mismo ng virus. Ngayon, kung halimbawa walang engine, ibig sabihin nun, puro RNA ng tao lang yung nakuha mo. Wala siyang infection. Pero pag meron siyang engine, yung nakuha mo is RNA ng tao at the same time, RNA din nung mismong virus. Okay, so pagka naglagay ka na ng primers, okay, mag-attach yun, hahanapin na kaagad nila yung engine, yung region for the engine. Now, after the first primer binds with the engine, ang mangyayari is yung enzyme called reverse transcriptase, i-extend niya ngayon yung strand by synthesizing a single-stranded DNA copy ng viral RNA, which is known as the complementary DNA or cDNA. So, gagawa siya ng DNA strand mula dun sa RNA na copy. Ang tawag dun sa enzyme, reverse transcriptase. And the RNA is then removed, tatanggalin na yung RNA, matitira yung complementary DNA. Now, that action is going to activate another primer na mag-a-attach dun sa complementary DNA na naiwan. Remember, wala na yung RNA mismo. Now, kapag nandun na yung primer, nag-attach na siya sa cDNA, complementary DNA, ang initiation is going to be done by attack DNA polymerase. Okay, so remember, kanina, yung nag-extend is called reverse transcriptase. Pero ngayon, ang mag-extend dun sa mismong dun sa next strand ng complementary DNA is the Taq DNA polymerase. Okay, so this produces a double-stranded DNA copy of the target region of the viral RNA. Ngayon, yung double-stranded DNA copy ng target region, mag-undergo siya ng actions of the polymerase chain reaction. Ano ba yung mga actions na yun? The first action is denaturation. The first process, denaturation, gagawin niya, yung DNA isa-separate niya ulit into two single strands. That's the first step. Second step is annealing. A-N-N-E-A-L-I-N-G. Annealing. Kung saan yung mga primers, magbabind na sila sa mga target sequences. Tapos yung ginawa rin kanina, yung last step is going to be extension. So, Taq DNA polymerase, magsisynthesize siya ng mga bagong DNA strand. Since yun, may bago ng DNA strand, mangyayari dyan, darami ng darami. Darami sila ng darami. Okay. So, ayun. So, dumami na yung copies mo. So, this process now goes on and on. Okay, each cycle doubles the previous number. Yung 2 copies, magiging 4. Yung 4, magiging 8, and so on. Now, a standard real-time PCR setup usually goes through 35 cycles. Actually, 35 cycles yun, na papalit-palit ng temperature. So, ibig sabihin nun, pagkatapos ng process na yun, yung buong process, around 35 billion new copies actually are produced. Okay? Yung viral DNA, nakikreate siya ng 35 billion times from each strand of the virus present in the sample. Now, in practice, the virus is typically detected in only about actually 30 to 45 cycles. Yan. Paano yan? I mean, okay, so nag-multiply siya, di ba? So, ano yan? Paano mo malalaman? It's because you will know it through a fluorescent probe. So, after halimbawang okay, so di pa naman natin alam, di ba, kung ano yung na-amplify, anong klaseng RNA. Although, yung RNA, alam natin na nag-bind na yung primer and all, gano'n, nakita na, we know that concept is going to happen, pero we don't have yet any way of counting, any way of quantifying what we're doing. So, you do that by adding a fluorescent probe. So, when you add it, it will allow the amount of target DNA to be detected in real time and get quantified after each cycle of PCR. Yung fluorescence, it's measured by the machine's computer, and then it's presented in real time on screen. The computer now is going to track the amount of fluorescence in the sample after each cycle. Kapag merong certain level na of light or fluorescence ang na-surpass, so this one, ibig sabihin nun, yung virus is present. Okay, so kapag wala namang fluorescence na nangyayari or hindi siya gaano nag-glimmer, pwedeng infected siya pero hindi naman siya gaano as infected as, mababa lang infection niya. Now, scientists also monitor how many cycles it takes to reach this level in order to estimate yung severity ng infection. Kasi kapag halimbawa, konting cycles pa lang tapos masyado ng matindi yung fluorescence, ibig sabihin nun, masyado mataas yung infection niya. Pero pag halimbawa, normal lang. Pero kapag halimbawa, matagal na marami ng cycles, wala pa rin nangyayari, ibig sabihin nun, negative siya. Ayun. So, it is clear na kung gagawin mo lang yung pinaka-importanting part ng buong process na to is the reverse transcriptase, RT-PCR. So, yung kaya nga tinatawag lang siya RT-PCR. Wala naman siya nilalagay, sinasabi na fluorescent probe or something. Ang pinaka-importanting part ng process na to is the reverse transcriptase polymerase chain reaction. This is Carlos, and it's another video for the Knowledge Catalog Education Solutions.